Next, we're welcoming Tamara Seredenina from a group leader at AC Immune, who will be presenting today on the development of fluid and PET imaging biomarkers for the detection of TDP-43 pathology. Welcome, Tamara.
Yes, good morning, good afternoon, everyone. Thanks for the introduction. I'm just putting full screen. Can you see the full screen?
Yes.
Perfect. So thank you very much for giving the opportunity to present our data in this exciting conference. And I will be telling you about our efforts in developing biomarkers, which is a nice transition from a fantastic talk that Katie just gave. This is my disclaimer, so I'm an employee of AC Immune, and part of this project has been sponsored by Michael J. Fox Foundation. Yeah, so as you all know, TDP-43 pathology is found in most of the cases of ALS, as well as in more than 45% cases of frontotemporal dementia. And Katie has just shown us a broad distribution of diseases that have TDP-43 pathology, including Alzheimer's disease, where TDP is found as co-pathology. So it's been transformative the way that TDP-43 aggregates were used in postmortem analysis, in the way it improved our understanding of the disease development and disease pathophysiology. And despite the fact that TDP-43 pathology was associated to neurodegeneration more than 15 years ago, we still don't have the markers for early diagnosis of TDP-43 proteinopathies. And this is due, at least in fact, to the low abundance of pathological TDP-43 species that can be found in extracellular space. And we at AC Immune, we have developed a broad panel of monoclonal antibodies against different epitopes of TDP-43, including phosphorylated epitopes, and explored different combinations of antibodies measuring TDP-43 phosphorylated species in biofluids with very limited success so far. So the conclusion was that we need an amplification step in order to visualize these pathological species. And this is how we came to develop and optimize the seed amplification assay, which will be the first part of this presentation. And in the second part of this presentation, I will be showing you the progress we made towards the development of TDP-43 PET tracer, which again, we're all now seeing the transformative potential of tau and A-beta PET tracers in the way to further improve the clinical trials via better patient stratification, focused recruitment. And in addition to that, TDP-43 PET tracer could enable development of combination treatment for these complex diseases. So our interest in TDP-43 biomarkers as a company lies in the fact that we are developing TDP-43 targeting immunotherapy. But of course, we hope that the efforts that we are putting into biomarker development would benefit broadly the community of ALS and neurodegenerative diseases as a whole. So we have developed an antibody called ACI 5891, which is a pan-TDP-43 antibody that binds into the C-terminal part, the low complexity domain of TDP-43 with very high affinity. And the characterization of efficacy of this antibody in various preclinical models was presented in this forum last year by Dr. Tariq Afroz, and now it's published in the Neurobiology of Disease. So very briefly, to summarize what this antibody does, it shows efficacy in vitro and in vivo by depleting patient-derived seeds to decrease templated aggregation. In addition to this, we've shown in two different models of TDP proteinopathies that this antibody is able to decrease phospho-TDP-43 pathology as well as spreading, and we demonstrated neuroprotection in one of these models. And in addition to this, the treatment with the antibody rescues phagocytic impairments that are observed in microglia from ALS patients. The mechanism of action behind this immunotherapy approach is to prevent the spreading of toxic TDP-43 species and to protect the unaffected parts of the brain from the TDP-43 toxicity. And there is a lot of evidence that TDP-43 spreading is contributing to the disease. There is evidence from human sample analysis in this image shown on the left-hand side, where you can see that TDP-43 pathology initiates in the focal place in the brain and then spreads via anatomically connected regions. And this has been shown for both FTD and ALS. And actually, the spreading correlates very well with the spreading of clinical manifestation of disease. And it's also been shown that CSF, the cerebrospinal fluid of ALS patients, has some toxic features contributing to neurodegeneration. So possibly these are the TDP-43 seeds that can be found in the CSF that could be contributing factor. So to further explore this, to further understand the extracellular TDP-43 species, we have used the seed amplification assay that was developed by Carlos Shallow in Giuseppe Legname's team, where they have shown that TDP-43 seeds can be detected in CSF of patients with genetic mutations or in FTD and in ALS. So we've taken this assay and further optimize it by changing the substrate. And the principle of the assay, which is also called RT-QuIC, is shown on the top right of this slide. So basically, we have a substrate that aggregates, and this aggregation creates beta sheets that then can intercalate thioflavin T, and we can evaluate the fluorescence of THT. And when we incubate this reaction with the TDP-43 seeds, the kinetic of aggregation shifts to the left. So you see much faster aggregation in this assay. So we have taken this concept and we got the samples through the NILS consortium, the ALS CSF samples in collaboration with James Perry. And we used these samples to estimate the aggregation kinetics in this assay. And the results are shown here. In blue, you see the control, healthy control CSF aggregation kinetics. And in black is the ALS sample. So you can estimate that all ALS samples have much faster aggregation kinetic as compared to most of the control samples. So this can be quantified in different ways. So one way of quantification is shown in the middle, where we are looking at how fast each sample reaches a certain threshold of fluorescence. And in the graph in the middle, you can see that ALS samples are seeding much faster than controls, about 2 hours faster. And then on the right-hand side, we can also look at the same data by overall levels of signal that is achieved within the linear part of the curve. And again, here you can see that there is much more fluorescence in ALS as compared to control. So this is very encouraging because, first of all, we have established and optimized the seed amplification assay. And we also expanded the previous findings from the sporadic patients. So now our assay also shows that the seeds of TDP-43 are present in sporadic ALS cases, which makes, of course, this assay very interesting as a biomarker in clinical trials. So encouraged by this data, we wanted also to address another point. So how can we be sure that the seeding is actually driven by TDP-43? And to answer to this question, we have incubated the CSF samples with our TDP-43 monoclonal antibody, ACI-5891. And so we incubated them before putting in the assay, mimicking the situation in the patient who would be administered with the therapeutic antibody. And as you can see on the graph on the left-hand side, this is the example from one case where you can estimate that once the samples are pre-incubated with TDP antibody, the kinetic aggregation shifts back to the levels of controls, blocking this seeding species. And then on the right-hand side, we're showing the quantification of a broader cohort of samples. And again, the gray bars, which represent the ALS samples, indicate that once the samples are pre-treated with the antibody, the kinetic of aggregation is slowed down. So this is very interesting, and it shows us two important points. So one is that actually it confirms that the seeding of in this assay is driven by TDP-43 seeds. And the second important point shows that the immunotherapy, TDP-43 immunotherapy, will be able to neutralize the seeding species present in patients' CSF. So we're very excited by this data. And now we're taking this assay into the next level of validation. We will be looking in a much broader cohort of patient samples, establishing the limits of quantification, understanding the longitudinal samples, as well as working on the specificity and selectivity of this assay. So stay tuned on this part. And now let me switch gears and tell you about our TDP-43 PET tracer program. AC Immune has a long experience with developing PET tracers. We have developed PI2620, which is a tau PET tracer, and we have also developed ACI12589, which is our alpha-synuclein PET tracer, the first tracer that can visualize alpha-synuclein pathology in living patients. And now by using the same approach and by using our therapeutic platform that's called Morphomer platform, we have also discovered TDP-43 ligands. And so these molecules derive from the Morphomer platform, which is a non-peptidic small molecule platform with the CNS drug properties for brain penetration. These molecules have conformational specificity, which drives their specificity towards pathologic species versus the monomeric or physiological forms. Our medicinal chemists can further engineer selectivity against different co-pathologies, as well as the pharmacokinetic properties suitable for brain PET imaging. So by using this platform, we've identified ACI19278 and ACI19626, for which I will show you now the detailed characterization. So this slide shows target engagement and affinity for our two selected molecules by a technique that's called classical autoradiography. We're using here FTLD-TDP cases with type A and type B pathology, as well as non-demented control. The images on the left are indicating the intensity that we achieve in autoradiography experiments, and you can estimate that there is no binding on control tissue and there is a strong binding on type A and type B pathology, which is completely displaced by cell flow, indicating and proving the specificity of binding. And when we quantify this signal, which is shown in the graph in the middle, you can see that there is a clear statistical differentiation of type A pathology across different cases that we explored, and of type B pathology as compared to control. For type B, it's in particular interesting for the 19626 compounds shown in the bottom. And for the second compound, we're further exploring this on a broader number of type B samples. And this is very exciting because, especially for this audience, because the type B pathology is the type of pathology we expect to see in ALS. Whereas type A pathology, beyond, of course, the FTD cases, it's also pertinent to late NAD. So this data show the broad potential of these ligands to visualize the TDP pathology across different diseases with TDP-43 pathology. And we have also used the same technique to estimate affinity, as shown on the slide of the images on the right-hand side. So you see the saturation binding curves for both compounds, with affinity in the range from 10 to 20 nanomolar, so very high binding affinity. And what's most important is the Bmax to Kd ratio that we can calculate using autoradiography technique, which is more than 40, which is a very good indicator of the binding potential and ability to see the signal then in human PET. So to further prove the specificity of the signal, We are using another autoradiography technique. This time it's high resolution autoradiography, where we are able, by using photographic technique, we're able to visualize binding at the cellular level. And you can see on the top row the silver grain, which indicate where the compound is binding, where the compound is accumulating. And the same sections are immunolabeled with phospho-TDP43 antibody. And you can see this staining in orange. So as expected, in control tissues, we don't have phospho-TDP43 pathology, and there is no binding of the compound. Whereas on type A and type B tissues, there is abundant TDP43 inclusions for the typical shape. And you can see that the compound is binding almost to every inclusion, as shown here by red arrows. So this is an excellent proof of the specificity of the signal. So the compound is binding to aggregated TDP43, and there is no obvious non-specific binding, which would be indicative of binding to something else or to physiological TDP. So very nice specificity. We have further explored binding by using orthogonal technique, where we're looking at the binding in sarcosome and soluble fractions from FTLD, TDP type A and type B. Here again, you see the saturation binding experiment with no binding in non-demented control, as expected, and very nice binding for both molecules on type A and type B tissue, with the affinity range from 10 to 50 nanomolar. Another important feature for PET tracer, of course, is selectivity. These are the molecules that will be used in elderly population that has often co-pathologies, so it's extremely important to differentiate binding, especially from amyloid beta and tau. So here you see the autoradiography experiment on Alzheimer's disease brain tissues. The images on the left bottom row are showing immunofluorescent labeling for A beta in red and tau in green, and phospho-TDP that is absent in this particular tissue, whereas the A beta and tau are very abundant. And on the top row, you see the total signal for a specific reference for A beta, tau, or AL2 TDP43 ligands. And in the middle, you can see the cell block. So basically, this data shows that this brain has very abundant A beta and tau pathology, which is visualized by the specific ligands, which are blocked by the self block, whereas TDP43 ligands 19278 and 19626 show no specific binding. So this is an excellent proof of selectivity, especially using the autoradiography technique, which has a resolution similar to what we expect in human PET. And to further explore this selectivity, we have also used high-resolution autoradiography, as shown on the right, with AD sections rich in tau pathology, as visualized here by thioflavin S staining of neurofibrillary tangles. And in the bottom, you can observe the PI2620 tau reference binding to tangles with these black neuronal bodies, whereas 19278 and 19626 show no binding. And another proof of selectivity. Next, we have also explored selectivity, again, by an orthogonal approach by looking into homogenates of AD and PD brain. So on this slide, you see 3 saturation binding experiments, where in green, this is a specific reference for amyloid, tau, or alpha-synuclein, showing a very nice saturation curve as expected. And our 2 molecules shown in blue and in red show no binding, neither on AD brain homogenates, nor on tau PHF, nor on PD brain homogenates. So excellent selectivity. And last but not least, another important feature for PET tracer is the pharmacokinetic profile. Here, we were able to radiolabel both compounds with the radioactive fluorine, which is an important feature for the future use in clinic. And we have measured PET pharmacokinetic profile in non-human primates following intravenous administration. These are wild-type animals, so they don't have TDP43 pathology. So what we're looking at on the left-hand side are the PET images of monkey brain with the top row showing MRI and middle row showing the 0 to 30 minutes, the uptake phase, and then the 30 to 90 minutes washout phase. And when we quantify the signal, which is shown on the top right, for the whole brain, you can estimate that both compounds go very fast in the brain within the first 10 minutes. They reach the peak, and both reach significant concentrations in the brain. And then the washout is relatively fast and complete. And then we wanted to compare the washout between the two compounds. And this you can estimate on the graph on the bottom, where the SUV values have been normalized to SUV max, which allows us to see the difference in washout with the 19626 shown in blue, showing a faster and more complete washout. And another feature that we also explored is that we noticed that 19626 has a more homogeneous distribution in the brain. So with this, we compared these two molecules and we selected 19626 for the future development in patients. So to conclude, we developed and further optimized TDP-43 seed amplification assay. We confirmed the presence of seed-incompetent species in CSF from sporadic ALS patients. We demonstrated that our antibody, TDP-43 antibody, can neutralize the seeding species, and we're further optimizing this assay to explore its full potential as a biomarker in clinical trials. On the TDP-43 PET side, we have identified two first-in-class TDP-43 ligands, ACI-19278 and 19626, that show low nanomolar KD, so very high affinity on FTLD-TDP brain tissue. They have potential to detect TDP pathology across various TDP-43 proteinopathies, including ALS and AD. They are selective over common co-pathologies, including A-beta, tau, and alpha-synuclein. They have clean off-target profile, but that I didn't have time to show you. So we also shown selectivity over MAO-A and MAO-B, which are the usual suspects for PET tracers in CNS. And in addition, the molecules have rapid brain uptake and fast washout in non-human primates. And as a next step, we're taking 19626 into clinical validation in first-in-human study, which is about to start in a year from now. So with this, I'd like to thank the fantastic AC Immune team involved in TDP-43 projects, as well as our collaborators from Mass General, James Berry and Steve Arnold, which were instrumental to obtain the samples from Niels. We also have collaboration with under JP&D consortium on TDP imaging. And part of the PET program was sponsored by Michael J. Fox grant. And I would like to have a special thank to the brain banks and the patients and families who donate their brains for the research, because it's really very important for us for these projects to work with the material which is as close as possible to patient situation. And I'd like to thank you for your attention. Be happy to take any questions.
Thank you, Tamara. We're running a little long, so I think we have time for a couple questions. It was a fantastic presentation, and thank you again for the very data-rich presentation. The program has potential for your own programs and also for advancing clinical research in ALS more generally. We have a couple of questions relating to the seed assay. One comment is that there's potential to purify the seeds from the CSF. Have you tried to do that work and to characterize the TDP-43 seeds?
That's a great question. That was a part of the task we had in our Target ALS grant. We did purify TDP-43 from human brain, from brain homogenates. However, from CSF, it's a much harder task, again, due to the low abundance. So we really need to have some kind of amplification. And so what we did for the seed amplification assay, which actually the data has been just published in Brain Communications, we did the electron microscopy analysis of the material that we can generate in the seed amplification assay, and we see clearly the fibril formation, beta sheet formation, confirming the seeding reaction. But yeah, with CSF characterization would be very interesting to understand the CSF seeds. So it would require some additional work on immunoprecipitation and further enrichment.
And I guess relatedly, if you can deplete, I guess, the CSF of the seed template, is that more effective than just co-incubating with the antibody at preventing amplification of the TDP aggregation in the thioflavin T assay? So not just blocking, but actually depleting and then seeing what happens.
That's a great question. I think this was addressed in Carlos Sciallo's paper, in the first seed amplification paper. The way we did it was really to mimic the situation in patients where the species, the seeding species in CSF will be captured by the antibody, which will be administered to patients, and therefore we believe it's more therapeutically relevant the way we perform this experiment.
And then last question, is there a time frame in which that assay system might be deployed, at least in an exploratory setting clinically, as a sort of biomarker? Do you have a time frame in mind?
I hope as soon as possible, but as you've seen, it requires quite some optimization. It's been working very nicely in our hands in our lab, but to use in clinical setting, of course, the assay requires more robust characterization, optimization, standardization. So we are working on this. This is the plan for 2024. So I'll be able to tell you more at the through mid 2024, towards the end of 2024.
Thank you so much for your presentation. I hope you'll be available for a few more questions later this afternoon.
I'll be happy to take any questions by email as well. Thank you.